Hello students, welcome back to the channel. So our today's topic is what we study in agricultural biotechnology. Whenever we are enrolled in agricultural biotechnology, we have list of topics to learn. Okay, we have a curriculum. So it starts with cell and will have different applications of agricultural biotechnology. Let's decode this. So we can understand this by keeping an example in our mind that what we do in agricultural biotechnology. So suppose if in a lab some scientist is doing experiment in a like developing a genetically modified variety, okay, of any crop. Okay, so this is just an example. So this in this through this flowchart we can understand this, that what things has to be done for it, like what we actually do in agricultural biotechnology, a basic of it. Okay, so it starts with cell, like this many things which you can see here in this flow chart and in the upcoming slide are the topics you have to learn in agricultural biotechnology. I am just giving an example of it, like through an example. So cell is the structural and functional unit of life. Okay, the basic unit. The cell contains, the center part contains the nucleus. Okay, the nucleus is having chromosomes, chromatin, and then chromatin, after that we called it as DNA. The genetic material here is the DNA. The cell is of two types, prokaryotic one and the eukaryotic one. Prokaryotic are the primitive cells. They lack nucleus. They don't have nucleus. But eukaryotic cell have nucleus. Chromatin, that is a thread-like structure, is made up of histone and DNA. Histone proteins and DNA. DNA is a loosely packed form of transcription, like form for transcription, used in transcription. Now, DNA, that is our genetic material. Then we can see the genes part, and then the genes, the promoters, exons, and the intron part. The chromosome is having a point known as locus where we can find the genes. The DNA, like replication, is a process where the DNA undergoes replication, where it forms different copies of itself, and after that mRNA formed, and after mRNA proteins we get. So DNA to mRNA, mRNA to protein is a central dogma process. The DNA to mRNA can be called as transcription, and mRNA to protein is called as translation. Gene regulation part comes after replication. In prokaryotes, we do it as by operons, lac-operon and tryptophanoperon. In eukaryotes, the transcription factors, enhancers are RNA. DNA repair mechanism has to be done by the following you can see here. Then molecular tools like PCR, electrophoresis, vectors, cloning. After molecular tools, genome mapping has to be done, genome sequencing has to be done. Genome mapping can be called as two type, genetic mapping and physical mapping. The functional genomics, which the examples are like RNA sequencing, microarrays, proteomics, marker development, QTL mapping, then marker assisted selection has to be done, then agrobacterium and through gene gun, genetic engineering and transformation step has to be done. After all these steps, we have to do the field trials and the regulatory approval. And after that, the application part comes, where the genetically modifying crops, genome editing and biofortification, all this has to be done. Okay, like all are done. Now, so this is the topic for today's class, and if you have any query regarding this, you can ask in the comment section below. Okay, so thank you, stay tuned.